Hi, everyone, and welcome to the third installment of Getting Started with Standards and Reference Materials in Microbiome. As promised in the last installment, this section is going to cover questions to ask. And this section specifically is going to cover questions to ask when establishing your microbiome pipeline. Just to quickly review the questions that we're going to cover, first we're going to talk about how accurately do my results represent the true nature of my sample. Following that, we'll talk about how consistent are my lysis and extraction procedures. After that, we'll ask which piece of equipment has superior performance. The next three questions shown on here we'll actually talk about in the next installment, which are questions to ask when you have an established pipeline and wish to troubleshoot it or fine-tune it. So starting off with the first question, how accurately do my results represent the true nature of my sample? In order to answer this question, because when you think about it, every sample out there has some true measurable nature, but we usually don't know what that is. In this case, we need a sample with a known true nature. The bioinformatic analysis can utilize this known true nature to say what we expect to see given our analysis method. It can also say what the expectations might be based on things like the manufacturing tolerance of that standard, since pipetting and measuring microbes out and that sort of thing are inherently imperfect processes. Based on the observed measurements versus the expected measurements, we can then say what our residuals or errors are and start doing some very standard statistical metrics using those. Now, one nice thing is for these known input standards, and in particular the Zymo mock microbial community, there is something called a MIC score, stands for Measurement Integrity Quotient, and that is a self-contained automated bioinformatics package designed specifically to analyze these known input standards relative to a reference genome and expected observations, and be able to generate a report giving you indications about your accuracy, as well as an overall score for accuracy. This Zymo Biomics mock community standard is often a very good place to start when incorporating a standard, and also a good standard to start using when establishing your microbiome pipeline. It provides a pretty simple method to identify some pretty major deficiencies that might come up, and be able to address them before you're in production and before you potentially start generating data and utilizing samples for experiments that you might then discover are flawed. Something you'll see a lot during the next two sections is going to be this general microbiome workflow. It's going to cover everything from sample collection and preservation, your first step. That's obtaining your samples and getting them to the lab somehow. Following that, you have DNA or RNA extraction, then library preparation, sequencing, and bioinformatics. In the case of trying to figure out how accurately your data represent the sample that you're looking at, you would just add a whole cell mock community into your extraction step and treat it as a sample. To analyze it bioinformatically, you would first start off with your raw reads in the form typically of a FastQ. You would then run an alignment-based pipeline, and this is where things differ a little bit from what you would typically expect. Typically, with microbiome, you want to do taxonomic assignment, but for these reference materials, you typically have reference genomes. These are genomes that are known and well assembled for microbes that are known to be in your reference sample. So in this case, an alignment-based pipeline is a really good place to start. Typically, I recommend BWA as a starting point for an aligner for short reads. And for longer reads, I typically recommend Minimap 2. These are good solid aligners and will typically get the job done for you. And this step in your pipeline should generate aligned reads. Following this, you can use a program called SAMTools, specifically its IDX stats function. And this can count the number of reads mapping to each reference genome. This can also report back the percentage of reads that were unmappable. And if you have a high percentage of unmappable reads from your standard reference material, that's really worth looking into. And while there's multiple causes, frequent causes of this are either poor quality of read or contamination of the reference material at some point during the process. Either one of those should be looked into to identify if there's a specific cause if you have an unacceptable percentage of unmappable reads. Another program to run on this is going to be FastQC or MultiQC. Both of these, while not specifically interested in microbial standards, can give you some general metrics on your reads, which is also important to get at this point. From this, you'll get read counts and read metrics, and you can go into your next step, which is going to be starting to give yourself results. So as I said before, FastQC and MultiQC give generic reports on the quality of the reads. Reads per genome can be plugged into Excel or some other data analysis package of your choice, but at this time, these are going to be some relatively simple results that you can then use in these data analysis packages to say what percentage of the reads came from each genome and what percentage did I expect. The percentage you expect are going to vary depending on the method used. For example, Shotgun will give you one expected percentage for a microbe, but a microbe that has many, many copies of its 16S gene, if you're analyzing by 16S, might appear to overrepresent itself by 16S. The MicScore package, however, can handle this part automatically and just generate a pre-formatted report for you, as well as several figures to help you be able to identify deficiencies or confirm a lack of a deficiency. From these results, you can start doing interpretation. First thing I always ask is, do the overall read quality metrics look good? This again would come from something like FastQC or MultiQC. Usually if the read metrics do not look good, I'll actually stop my analysis at that point, just because I'm not going to be able to get a good interpretation of how I performed on the standard with poor quality overall reads. If the read quality is poor, that's likely to indicate an issue with your sequencing or library prep, but less likely to indicate an issue with your standard or your microbiome pipeline, although that may be the issue as well. But either way, if you get poor quality metrics overall, the best thing to do at that point is just stop everything and find out what went wrong with the sequencing or what caused these poor quality metrics and address that issue first and foremost. Assuming that your read quality metrics overall look good, then you can start asking, how much error was there that could not be accounted for by manufacturing tolerances? Or in other words, how closely does what I recovered from my microbial standard recreate what I know went into my microbial standard? And again, this is where the MicScore package might come in handy, because if you run this package, a lot of this will be answered for you automatically and generated into a report. Now I keep talking about the MicScore, so the question comes up of what is the MicScore? So this is measuring accuracy relative to a standard. The way we do this is each dot here represents a different microbe, and we just did a very quick transformation. So each dot here, we have a microbe at each point on the x-axis, and the y-axis value represents the percentage of expected that was actually observed. So for example, if we see a score of 120 for a microbe, that means we observed it 120% of what was expected, or we observed a little bit more of it than was expected. If it's right on that black line, that means that it was 100% of expected, or we observed exactly what we expected to observe with it. You'll also notice that there's two thinner blue lines, and that accounts for plus minus 15% manufacturing tolerance. And we know that this manufacturing tolerance has to be accounted for because pipetting and measuring microbes, again, is an imperfect process. So anything that is within those two blue lines is as accurate as can possibly be given the inherent inaccuracies of the production of the standard. All we do following that is we measure residuals, or errors, or deviation from the expected. These are all different ways of saying we're measuring error. Only in this case, rather than measuring error relative to 100% of expected or a theoretical perfect, we're actually taking into account the reality that there is a manufacturing tolerance, and we're measuring to the range of our manufacturing tolerance. Or in other words, we're saying how much error is there that cannot be accounted for by the inherent inaccuracy generated by manufacturing. We take the tried and true statistical formula of root mean squared of errors and apply it to these errors and subtract it from 100, since we're working in percentages. So in theory, if every single dot was inside that band of manufacturing tolerance, we would be able to assert that everything we measured was as accurate as possible given the inherent inaccuracies of the microbial standard. And that would give us a MicScore of 100. And that's a perfect 100 MicScore. In neither case here do we see that though. We see some measurements that come just even a little bit outside of that line, or sometimes even way outside that band. So for the example of a good process, we see a score of 93, and I typically like to interpret these as I would a grade on an exam in a high school class. So high 80s or 90s, it's doing pretty well, especially consistently in the 90s. Typically, if a score is in the lower 80s, especially if it's consistently in the lower 80s, or 70s, I would say there's definite room for improvement and probably some fixable deficiency there. Once the scores are dipping down into the 60s and below, I would say there's significant deficiencies and they probably need to be addressed. And you can clearly see that in this case that I show where the score is 30, and we see that everything is way off of expected and way outside that band of manufacturing tolerance. Now this takes us to the next question. How consistent are my lysis and extraction procedures? And it turns out that this is a relatively simple question to answer, given that this fundamentally requires us to answer the first question, but just answer the first question a bunch of times in a row. A pretty common statistical metric used to look at this is called the coefficient of variation, and this can actually be done with a Mike score. However, while I won't get into the hardcore statistics of it, measuring a coefficient of variation requires that a value of 0 means something. The Mike score value of 0 is actually not what would be considered a statistically meaningful 0. However, the Mike score of 100, which is essentially a perfect score, is statistically meaningful because that's actually making an assertion of 0 error that can't be accounted for by manufacturing tolerance. This actually gives us a very easy way of doing a quick transformation by just taking 100 minus the Mike score. And when we take 100 minus the Mike score, that gives us a meaningful 0. So just applying that simple transformation gives us values that are useful in calculating a coefficient of variation. And lower coefficient of variation typically means more consistent performance and much less error overall. This can also be carried out using a complex reference material such as the true matrix fecal reference to provide a repeated consistent sample. This gives us the advantage of being able to run our pipeline and compare it to a more realistic sample, especially if your typical sample type is fecal. However, the analysis methods you might use for this are going to vary and likely be a little bit more complicated than just running repeated Mike scores. In order to do this analysis, we would add either the whole cell mock community as before to our extraction step, but if we're going to use a complex reference material, we would add that instead here. The initial bioinformatics workflow, I'm not going to go over step by step because it is actually the same workflow as I showed for the previous question. It's just that we're going to run this workflow several times over. And along with measuring things like coefficient of variation, we can also just do a process control chart. And in this case, the x-axis here shows just different runs over time. The y-axis shows the Mike score for each run. And this is the kind of image we want to see. We're consistent. We have just a little bit of normal variation. We're constantly keeping ourselves in the high 80s or 90s, and if I saw this coming out of my lab, I'd be very happy with the performance. The kind of thing we don't want to see looks more like this. And this is the advantage of running standards with every single prep, is you can very quickly pick up on this kind of situation. So we have the first same 10 runs as before, where we have very good performance. Starting with run 11 and definitely into run 12, we're seeing a decline in performance that actually starts declining more and more with each run. Now this is based on a real scenario that I saw, and really starting with, in this case, the 12th run, an investigation was started into why the performance was declining, and nobody could quite figure out why the performance was declining despite our best efforts. Except on run 15, that lysis did not go to completion, and in fact there was a major malfunction with the bead beater where it essentially catastrophically failed and had to be serviced. At the time of service, it was determined that a bearing had failed, and that bearing was likely failing over several runs, which explains why we had a consistent failure. Maintaining a process control chart like this also let us kind of go back in time and be able to make some pretty confident assertions, such as run number 11 may or may not have been affected by the declining quality from the failing part. We actually can't say because it's in the normal range, even though it is on the decline. Run 12 was most likely the first run where we can say, yeah, this was really affected. And beyond that, we're definitely in the decline, no question about it. And just for safety's sake, since run 10 looks like it was clearly not part of the decline, we actually were able to say, let's repeat run 11 and everything beyond that, just to be sure that we don't have any data that was affected by the failing machine. You can also use your own metrics, and again, this could be more complicated and you might have to come up with your own method for doing this to compare a more complex material such as the true matrix fecal reference. In this case, we're looking at some of the more common taxa in there and just generating taxonomy plots and seeing if they look similar. And while taxonomy plots aren't the most informative if you have large differences, it is usually pretty easy to see that you have a good degree of similarity between samples. Moving to our next question, we can ask which piece of equipment has superior performance. And it turns out this is also now similar to previous questions, and again would require a known input reference material. In this case, a known input reference material would be much more useful than anything complex or unknown. And we can use this to both compare different settings on a single piece of equipment. So if we know that we're going to be using a specific bead beater, we can use this to figure out which settings are optimal on that bead beater to get us the most even lysis, the most accurate picture of the input sample. We can also use it to compare between pieces of equipment to determine which one has better performance at some given setting. And in this case, you would try different pieces of equipment at different settings or maybe at different run times. For this one, just as before, we're going to add a whole cell mock community to our DNA or RNA extraction process, and we just chart out different settings in different machines. So in this case, for example, with bead beater 1, and for all the following figures, we're going to see the mic score generated for a triplicate of samples on the y-axis, and the time that we run the bead beater on the x-axis. In the case of this bead beater, we can see very clearly that we've hit a plateau by about 3 to 5 minutes of bead beating. Running it out to 10 minutes doesn't give us improved performance, and in fact, we are, by these 3 to 5 minute period, we plateau out in the high 80s or 90s. Again, giving us a very acceptable performance, and now we know running this machine 3 to 5 minutes is going to probably give us a consistently good lysis. For bead beater number 2 here, and you'll notice we have a slightly different x-axis on a different scale, 10 minutes on this machine gives us highly inconsistent performance, with one sample looking pretty good and 2 samples looking pretty unacceptable. Running this machine out to 40 minutes, though, starts giving us good performance. And in fact, by 30 minutes, and especially by 40 minutes, we're seeing consistently scores again in the high 80s or 90s, which is where we want to see them. So this machine is also acceptable, so long as we extend the bead beating run a little bit longer. Bead beater number 3 here, we pretty much start low and we stay plateaued out at a very low number, never really getting above 40 or 50. Actually never really getting far beyond 30. So this is a machine that I would say is probably not going to be acceptable to us. And that brings us to the end of this section. I hope you enjoyed it. Please come back next time for the second set of questions, which are questions to ask when getting started with when you have an established pipeline and want to do some fine-tuning of it. Thank you.